Here are the learning outcomes for that previous video, and this next video is going to connect very nicely with the propagation, the conduction down the axon. So the ones we just covered are describe the physiological process in conduction of an action potential, including the types and locations of the ion channels involved. So these two types of ion channels that we talked about. And then we also distinguish between the absolute and relative refractory periods and the basis for each. This is due to inactivation or the hyperpolarization. And lastly, we explain the impact of these two refractory periods on activity of the neuron. So in terms of the amount of stimulus that will be needed to generate a new action potential. The last thing about conduction goes back to myelination that we talked about last week. So first of all, action potentials are all or nothing. So this first picture here shows a neuron if it didn't have any voltage-gated channels in it. This is not a real situation, right? But this really emphasizes the importance and the meaning of excitable membranes. Excitable membranes, excitable cells, muscles, and neurons have voltage-gated channels, which allow them to respond to voltage and have an action potential travel down the entire length of an axon or throughout an entire muscle cell. If you didn't have those voltage-gated channels, you'd have just a normal cell, which are, some of them are really cool too, of course, and you wouldn't be able to have this action potential travel down the whole axon without decaying or getting less, right? Decaying means getting less, degrading. So if you have voltage-gated channels, you're able to have voltage-gated ion channels that regenerate that electrical signal wherever they are located. So the flow of sodium ions in due to new voltage-gated ion channels being opened, that positive feedback. Depolarization causes more voltage-gated ion channels to open. That is due to voltage-gated ion channels, and that allows the action potential to propagate down the length of the entire axon. However, this is fairly slow because we've got to go this entire length of the entire axon. Unmyelinated neurons do this, and these are, a lot of the neurons in your body are unmyelinated. They're fine, they do their thing. However, if we want to be able to have this action potential conduct faster, that's where we have myelination, these myelin sheaths around the neurons. So myelin sheaths I talked about last week, they're due to cells that surround the neurons. These Schwann cells in the peripheral nervous system, oligodendrocytes in the central nervous system. And this is basically a cell surrounding the neuron that provides insulation. And this myelin sheath is made up of actual layers of cells, so it's fatty, and so it's going to be insulating. This allows the action potential to go faster. So it speeds up conduction. A large diameter neuron will do a similar thing. So this is called, we jump from here to here, it's called saltatory conduction. And I'm going to draw this on here. So again, we've got our neuron here, and then we're going to add some myelin. So this myelin sheath surrounds the entire neuron, but I've kind of got a cross-section cut here, longitudinal section cut, where we are able to see the inside of the axon. And what's happening is we've got sodium flowing into the cell, as you know, and then spreading out, diffusing once it's inside the cell. This insulation here allows sodium to spread farther than it would if there weren't insulation there. Because of that, that sodium can flow all the way down to the next node of Ranvier. That's what is here, a node of Ranvier. This yellow stuff is our myelin. So the myelin being there allows, okay, when this sodium gets here, what happens? It opens voltage-gated sodium channels. Sodium flows in here, diffuses both directions, This way though, doesn't matter because these channels are inactivated. When sodium flows down this direction, it's going to cause the channels here to open. So by that mechanism, we're able to jump from node to node. The signal jumps because of the insulation. The ions are actually able to move farther within the cell. That's called saltatory conduction. I have it right there. I don't need to write it. Saltatory conduction. Saltatory refers to jump. So saltar in Spanish is jump. Okay, so that means it moves faster because you don't have to open as many ion channels. It's kind of like using the tab button on your Word document instead of spacing all the way across your entire screen. Okay, so what we've just done is explain how axon diameter, so that was just the fact that larger diameter axons allow signals to travel faster than smaller diameter axons, and myelination affect conduction velocity, and compare the conduction in unmyelinated versus myelinated, faster versus slower. Then explain the role of myelin in saltatory conduction.